Transpiration of Oregano. Having placed a focus on transpiration in plants, we decided to name our investigation the effect of light intensity on the rate of transpiration in oregano. Within this, we discovered the direct relationship to the transpiration rate of one stomata in milliliters per hour. The independent and dependent variables are light intensity and rate of transpiration, respectively.
The purpose of this lab is to figure out how light intensity affects the rate of transpiration in oregano, and from this, what the transpiration rate of one stomata in milliliters per hour in oregano is. By placing the two plants in different areas of different light intensities and weighing them at regular intervals, the information can be used to calculate the rate of transpiration in one stomata. The data was then compared to the rate of transpiration in the control group to determine how light intensity affected it. The plant that was placed in the jug transpired 0.748 times less than the control group, while the plant placed under the light transpired 1.08 times greater than the control group.
The transpirational pull in plants helps bring water up through the roots and to the leaves. It sucks water up through the stems, similar to how a beverage moves through a straw. In the leaves, some of the water is used for processes such as photosynthesis, but the excess water is released into the atmosphere through openings called the stomata. The driving force for transpiration is the difference between water potential in the soil and atmosphere surrounding the plant. As a result of this difference, there is a gradient that forces the water to move into areas with less water. As the light intensity increases, the opening of the stomata increases as well. Therefore, if oregano is placed in an area with a higher light intensity, it should have a higher rate of transpiration as well. As opposed to the control group, where the plants are placed in an area that is exposed to regular school light bulb intensity, the plant placed in the dark will have a lower rate of transpiration. Conversely, the rate of transpiration of a plant placed in a high light intensity will have a higher rate of transpiration.
From our research, we reached the conclusion of proposing a question involving how different intensities of light can affect the rate of transpiration in an oregano plant. From our background knowledge, we believe that if the amount of light increases, the transpiration rate will also increase because water evaporates faster through the leaves' pores when there is a higher presence of light.
Please take the time to read through the procedures.
So what exactly went down in this lab? Knock knock. Who's there? Leave. Leave who? Leave me alone. Why did the chef add extra oregano to the sauce? 'Cause he was making up for lost time. Here shows the data collected from the controlled run of the experiment. It can be seen that Greg lost about 2.3 grams of his mass every hour, while Reagan lost about 2.1 grams in the same time interval. Now we see the gathered data from the experimental test. With Greg placed in an area with an absence of light and Reagan under the fume hood with light projected upon her, it is revealed that Reagan lost more mass over time. This indicates that she had a higher rate of transpiration compared to Greg, who was in the dark. These pictures were taken during the readings of both oregano plants and the control test. We added 6.5 grams to the values because the Ziploc bag was not yet attached at this point. Next, it was necessary to take off the leaves of the oregano plants in order to make the stomata peels and view them under the microscope. While observations were being made under the microscope, each member made counts of the stoma in 3 field of views. The average was then calculated, and these results indicate that both plants have a higher average number on the backside of their leaves. Take a moment to pause the video to look more closely at the data.
To find the rate of transpiration per stomata, we first found the mass of all the leaves on each plant. In our first plant, the mass of all the leaves was just over 23 grams. Then we took one leaf and cut it into 1 square centimeter and measured that at 0.022 grams. By dividing these two numbers, we were able to find the entire surface area of the plant at 1,057.95 centimeters squared. Next, we found the number of stomata on both the front and the back of the leaves. For the front, we took the average number of stomata divided it by the field of view, and through dimensional analysis found the total number of stomata. We repeated this process for the back of the leaf and added these two numbers together to get the total number of stomata for the entire plant. We repeated this entire process on our second plant. After finding a surface area of 1,285.26 centimeters squared, we used dimensional analysis for the front leaves and for the back sides of the leaves. Finally, we added these two numbers together to find the total number of stomata for our second plant. Our second set of calculations had to do with finding the average rate of transpiration per hour in each plant. To do this, we measured the amount of water released per hour and averaged it up for a total of 5 hours. We did this for both the control and experimental groups. Then we divided these averages by the total number of stomata to find the average rate of transpiration per stomata. Within our control group, the average rate of transpiration per hour of the entire plant was 2.34 grams, and our other plant was 2.15 grams. In our experimental group, one plant averaged 1.75 grams per hour, and another averaged 2.35 grams. On the right side of this diagram, you can see our first plant, Greg, who was placed in a darker condition, showed a 0.748 times less transpiration rate than the control group. In our other plant, however, Reagan, she showed a 1.08 times greater rate of transpiration after being placed in the lighter environment. These results show the amount of grams of water that each stoma released per hour in each plant. It is easy to see here that in the control group, Greg released a lot more water than in the experimental run. In contrast, in the control group, Reagan released a lot less water than she did in the experimental run. By calculating the transpiration rate of the entire plant per hour in both the experimental and control groups, we were able to see that the whole plant will lose more water in the lighter environment and less in the darker one. Calculating the entire surface area and the number of stomata within the entire plant helped us to find the transpiration rate per stomata per hour. Our results showed that the amount of water transpired in the lighter conditions was 1.08 times greater than that of its control group. This showed that the plant growth was more effective in the lighter conditions. The experimental plant was able to grow better in this light box than it was in the classroom. On the other hand, the amount of water transpired in the darker conditions was 0.748 times less than its control group. This means that the plant was not able to grow as well in the darker conditions and release enough water. These results supported our hypothesis, which stated that the lighter environment was more ideal for oregano growth and that each stoma would release more water per hour in the lighter conditions.
So during this lab, when wrapping up the oregano and dealing with it, some of its leaves broke off and fell. In addition, we might have overwatered one of the plants, and also, in the end, some of the baby leaves remained on the plant and not all of them were picked off. These are all possible sources of errors as they could give inaccurate mass readings. These inaccurate mass readings would affect our calculations, causing our results to turn out differently.
Oregano, oregano, wherefore art thou oregano?